and please take it away.
Okay, well, thank you, Anita, for this wonderful introduction. And thank you all for joining, and welcome to today's webinar titled Charting the Brain's Connections in Interactive Connectome, the Rat Hippocampal Formation, Parahippocampal region, and retrospinal cortex. We will try our best at making it a dual presentation where both Niels, that's me, and Natalie will speak about our work. The first explorations of anatomical brain networks started more than 100 years ago by Santiago Ramón y Cajal, and during the last century, the knowledge of the brain connections has expanded significantly. Many scientists now agree that knowledge of the anatomical networks lies at the basis of understanding brain function, and the amount of network knowledge is important for the level of detail at which we can explain network function. In other words, the more detailed our knowledge about brain networks gets, the better we are capable of inferring network function. A problem that many neuroscientists face is that the details about brain connections are hidden in the literature by, for example, nomenclatures that can only be compared when making a huge effort to understand the implication of the different terminologies that are used. To contribute a solution to this issue, we, Natalie, Niels, and Menno Witter, developed this interactive connectome of the rat hippocampus, parahippocampus, and retrosplenial cortex, containing all known connections between these regions based on all available literature. A brain's connectome has similar useful features as a roadmap. For example, maps can provide insight into the movement of traffic. To understand more about the traffic situation in the left image, it would be useful to have a road map. The map allows you to see, for example, how this traffic continues and can move out of the city. Adding extra functional knowledge to a map, for example the traffic light, perhaps explains why the cars in the left image are waiting. When such functional details are added to a map of connections, this increases its explanatory potential. In the world of brain connections, the buzzword is currently the connectome. But what exactly is meant by connectome? Olaf Spons came up with the following definition. A connectome is a comprehensive description of the network elements and connections that form the brain. Since many scientists trust that this approach is fruitful, several concurrent attempts are being made, like the Brain Connection Management System and COCOMAC. We feel that our temporal lobe connectome has several noteworthy differences compared to other connectomes. It contains more detail about the three-dimensional position of connections than is available in other databases. The connectome displays all connections presented in the currently available literature, which makes the connectome a picture of the current state-of-the-art knowledge. The connectome is freely available as an interactive visualization, and so far the updates have been accompanied by extensive reviews of the anatomical literature.
Before presenting our connectome, I first want to walk you through the steps of how it was created. The process of making a connectome can be summarized into 3 steps. Step 1, searching for literature. Step 2, reading and verifying the literature and collating. Step 3, interpreting and compiling the connections. These steps will be explained in greater detail in the next slides. For the first step, the literature search, I will give an example of the connections of the retrosplenial cortex. Typically, a person would search PubMed like this. Enter a search term like retrosplenial cortex. You add what you want to find out about this first term. In this case, it's anatomy. Finally, you add the species you are interested in our case, the rat. Instead, thanks to Ingrid Riphagen, we used an elaborate PubMed and base search. This search term for anatomical regions is significantly larger, as well as the search term for brain connectivity. Finally, a search term for the animal of our current interest, the rat, added. This type of literature searching provides you with a vast list of citations, which more or less all reporting on anatomical connections. If it's relevant to you, feel free to use our query for your benefit. It is published on our website temporalobe.com. The combined searches in PubMed and in Embase resulted in more than 800 unique papers for the retrospinal cortex search. We now move on to step 2. From the 800 papers we found, we first selected the papers of interest on the basis of 3 criteria. The papers must provide information on or intracellular filling, use healthy, untreated and genetically unaltered rats, and citations must be written or translated into English or another language using Roman print. Out of the 800 possible targets, only 46 citations contained relevant information about the retrosplenial connections, hippocampal formation, and to the parahippocampal region of the rat. Finally, each connection was verified for accuracy by a second collator, and only when approved it was entered into our relational database. When we created a relational database in Microsoft Access, the left image displays the tables and their relations. On the right side of the screen, you see the data entry interface. In addition to brain connections, we also recorded the method for determining a connection in our database. For example, details about the track tracing methods like the dye and the injection methods which was used in the paper. We now move on to the third step and last step of the connectome development. This step involves querying the database for all connections between all areas. We choose to export this data to an Excel file, and each Excel file contains all the reported connections across the literature between an area A and an area B. In the next slide, we will look into such an Excel file and explain how this data is processed further. We will focus on the connection sheet for area 30 to area 29C of the retrosplenial cortex. All connections for a given region are so gathered into the Excel file, and on the slide you see such an overview of each individual record that describes the connection between the retrosplenial area 30 and the retrosplenial area 29C. In the first column, the article ID number in the database is noted, and in this case there are only 2 papers visible in this part of the Excel sheet, but there are of course more. In the fourth column shows the from area, which in this case is the area 29C. Oh, area 30, I'm sorry. In the seventh column it gives the to area, which is in our case 29C. Next, the connections that resulted from the database query were considered to be included in the connectome only if they met certain criteria. When it was clear where anterogradely filled terminals or retrogradely labeled cell bodies were located, when the location of the injection site was clearly described, if the injection site did not include multiple brain areas or fiber bundles, also lesion studies were discarded, explicitly reported non-excitatory projections were excluded, And last, connections to the contralateral hemisphere were also excluded. From the connections that are included in the connectome, we automatically generate these kinds of summaries, and then decide which connections to draw based on the combination of anterograde and retrograde tracing results. In the next example, we will show how this process of combining track tracing results across experiments is performed using our knowledge of connections concerning the retrosplenial areas A30 and 29C. We will show some parts of our connectome to illustrate this example. In this case, an anterograde midrostrocaudal A30 injection results in terminal labeling in layer 1, 2, 3 and 5 of the rostral area 29C. To accurately determine the originating layers of this projection, we look for the existence in the literature of a retrograde tracer that is placed in rostral area 29C. This retrograde labeling experiment reveals the layers of origin of this projection from midrostrocaudal A30, layer 2, layer 3, 5 and 6. The limitation of the approach is that we cannot tell if each of the originating layers project to all the target layers or only to a subset. For example, does layer 2 project to layers 1, 2, 3 and 5? Or could it be that it only projects to layer 1? Currently, our connectome displays projections from all layers to all layers, since more detailed information is virtually absent in the track tracing literature. Only when single cells are fully traced, one can describe layer specificity of projections. Finally, we draw these connections in Microsoft Visio, a charting tool, and convert the result to PDF. After a little programming, an interactive PDF connectome is born. Through these 3 steps, we constructed an unbiased, interactive, species-specific connectome of all reported brain connections for a selected number of brain regions. There are 2 features of our connectome we want to emphasize. First, interactivity is a special feature of our connectome, since most are not. Take for example this beautiful but non-interactive diagram you see on the right side of your screen from Fernando de Silva. Second, the connectome is accompanied by a website, which you can find at temporallobe.com. It hosts free online researches such as a reference search, our PubMed queries, and the connectome itself, of course. Thirdly, in case you like to know more about hippocampal and parahippocampal anatomy, I would like to direct you attention to this freely accessible Hippocampus Atlas. The Hippocampus Atlas and our connectome use the same color coding of the areas, such as the orange area CA1, which you can see on the screen now. In the current version of the connectome, 3 brain regions are included, and those are the hippocampal formation, the parahippocampal region, and the retrosplenial cortex. In the next few slides, I will explain how these brain areas are represented in the connectome. I will make an effort to explain how 3-dimensional position is defined within the anatomical structures that are included in the connectome and how this is displayed in our interactive visualization. To define 3-dimensional position in the brain, 3 axes are defined for each structure. Functional differences exist along these axes, and therefore a connectome should incorporate such positional information. Here you see a lateral surface view of half a rat brain. In this view, position can be determined along the dorsal to ventral axis, along the rostral to caudal axis. The yellow shaped C-shaped structure represents the hippocampal formation. Note that the hippocampal formation has a bent dorsal ventral axis, which is also called the septal to temporal axis. In order to determine 3D position in the brain, we also need the third axis in addition to the rostral to caudal and the dorsal to ventral axis. This axis is exposed when splitting the brain with a horizontal cut, and when looking on the cutting plane, the lateral to medial axis of the brain is exposed. First, let's take a closer look at the subdivisions that are found within the hippocampal formation. The highlighted structure you see at the moment is called the dentate gyrus. In the dentate gyrus, the medial to lateral axis runs from the exposed on your left side to the enclosed plate on your right side. In the next view, there it is, the CA3 and CA1 fields, both part of the corneomonas, are highlighted. Note that in the current version of our connectome, CA2 is not present. This will change in our next update of our connectome. Position in the medial to lateral direction is based on the proximity to the dentate gyrus, and this is called the proximal to distal axis. For the subiculum, the proximal to distal axis is also used. Finally, one can discern cortical layers in each of these areas. In a connectome, all this anatomical information needs to be described. In our connectome, sub-areas of the hippocampal formation are displayed by boxes. From left to right, one can see the dentate gyrus, the CA1, the CA3, the CA1 and the subiculum. To understand what is displayed in our connectome, we will discuss hippocampal area CA1 in greater detail, and we will start with the horizontal slice I showed previously. We've already covered how anatomists define 3D position of CA1 in the rat brain, but how can one capture that information in a two-dimensional figure? Before being able to answer this question, one has to understand how anatomists describe brain connectivity in scientific reports. Unfortunately, the literature is very unsystematic and information about brain connections is presented at varying levels of spatial detail. For example, one could find in the literature that CA3 projects to CA1. This is a very generic description when compared to the projection from the stratum pyramidale of distal CA3 to the stratum radiatum of proximal CA1. To include all possible levels of spatial detailedness of a description of brain connections in our connectome, we created a representation of CA1 with varying levels of spatial detail. To understand this, first divide the area box representing CA1 into 4 quadrants. Next, give each quadrant a number. Quadrant 1 describes connection at the highest level of anatomical detail. In the white boxes, we find the layers of area CA1, which are stratum moleculare, stratum radiatum, stratum pyramidale, and stratum oriens. In addition to the proximo-distal axis, also the proximo-distal axis of CA1 is visible. Finally, information can be displayed along the septotemporal axis of CA1. In order to display this in the brain, we have to look into a coronal section. Information about the septotemporal axis in the CA1 area is represented along the vertical direction. If information about 3D position of a connection is incomplete, the lines that are drawn in the connectome will originate from quadrant 2, 3 or 4. Quadrant 2 describes connections of CA1 at the level of layers and along the septotemporal axis. Quadrant 3, on the other hand, describes connections of CA1 along the proximodistal axis and layers. Quadrant 4 describes connections when only layer information is present. Without going into the anatomy much further, I would like to point out that in a similar way as in CA1, the presubiculum and the parasubiculum are presented by blue boxes, and the entorhinal cortex is presented by green boxes. Furthermore, the perirhinal and postrhinal cortex are both presented by purple boxes and by one blue box. In the next slide we see a mid-sagittal surface view of half a rat brain again, and the retrosplenial cortex is a neocortical structure situated in the midline of the brain. It arches around the dorsal caudal half of the corpus callosum in the rat. Position within the retrosplenial cortex is also defined by three axes. We have the rostral to caudal axis, the dorsal to ventral axis, and the lateral to medial axis, which is not shown in this slide. According to the nomenclature of Vogt, the retrosplenial cortex is subdivided into four regions: area 30, area 29C, area 29B, and area 29A. Most studies do not separate between area 29A and 29B. And to be able to cover more of the connectivity literature without having to make too much assumptions, we included a region 29AB as the unseparated version of area A and B. This is how the retrosplenial cortex looks in the connectome. Most left is area 30, in the middle is area 29C, and on the right there are boxes for 29A and B, and a separated version of the area 29A and area 29B. Note that for the latter regions we limited the spatial positioning system to consist of layers and subregions only, since too little information was available about rostrocaudal and dorsoventral divisions.
Up to now, we have focused on the implementation of our connectome. We now want to point out some of the challenges that we faced when making this connectome. It is good to be aware of the current limitations of our approach, and we will go through each of these challenges. Reporting on brain connections with a century of literature is difficult because the nomenclature and conventions changed over time. Although nomenclature issues exist for almost all brain regions, we provide here an example for the retrosplenial cortex. Kriek was the first to delineate the retrosplenial cortex in the rat. Kriek divided it into a ventral subdivision, area 29B, and a dorsal subdivision, area 29C, based on the nomenclature of Brodmann. Brodmann subdivided the retrosplenial cortex in rabbit and dubbed it area 29. Brodmann divided area 29 into area 29A, B, C, and D. Many more parcellation schemas appeared throughout time. In our connectome, we selected the nomenclature of Vogt for retrosplenial cortex. We provided this Rosetta table of nomenclatures, which is a necessary translation tool that lists all available nomenclatures, in this case for the retrosplenial cortex. Challenge 2 relates to the question how to differentiate between weak and strong connections. Most anatomical reports contain subjective descriptions of the strengths of projections. Such subjective reports are impossible to quantify, and therefore such information cannot be incorporated in the connectome. We recognize that in reality, differences in connection strengths exist. For example, area 29AB connections to the medial entorhinal cortex terminate in both superficial and deep layers, indicated in the figure by connectors to all layers. Clear differences between connections to superficial and deep layers are known to exist. There's a dense terminal labeling in layer 5 of the medial entorhinal cortex, whereas terminal labeling in layer 3 is very light. In the connectome, these connections appear of equal strength. Yet, on the basis of the literature, it would be reasonable to suggest that the main effect of area 29AB on the medial entorhinal cortex is mediated through layer 5, not through layer 3. Another challenge is how to interpret negative information. There are two types of negative information. Data can indicate that a connection between area A and area B is very unlikely to exist, or a connection between area A and area B was not reported, but may exist. An example, area 29C and area 29AB both project to the subiculum, whereas data on the existence of a projection from area 30 to subiculum is lacking. Should one now conclude that a projection from area 30 to subiculum is not present in the rat brain, or could it be that this projection exists in the brain but has remained undetected, or was detected but simply not reported? Once sufficient high-quality reports of existing and non-existing connections are available, users of a connectome can be reasonably certain that any particular connection does or does not exist. Currently, caution is required. Challenge 4. The inclusion of incorrect data. Although it is assumed that researchers aim to report as accurately as possible the results of their track tracing experiments, one cannot rule out the possibility that brain connections are reported that do not exist. The next one is maybe a surprise. It is very important that claims in articles are supported by sufficient data to confirm an observation. And sometimes a mismatch exists between textual interpretation and the provided evidence. To conclude this talk, I will give an example of an inferred experiment based on our connectome. For this thought experiment, we will try to infer a functional subdivision of area 29C on the basis of known connectivity patterns. In the track tracing literature, traditionally an emphasis is put on connections running from rostral area 29C to septal subiculum, and from caudal area 29C to both the septal, intermediate and temporal parts of the subiculum. Although we noticed an emphasis in the retrospinal track tracing literature on this topography, for us it was too aspecific to make functional inferences for area 29C. However, upon close inspection of the connectome, a striking topography is apparent in the output projections of subiculum to area 29C. The proximal subiculum projects to the rostral part of area 29C, and the distal subiculum projects to the caudal part of area 29C. This topography could be of functional relevance, since it is known that the lateral entorhinal cortex projects to the proximal parts of the subiculum, whereas the medial entorhinal cortex projects to the distal parts of the subiculum. A lot of emphasis has been placed on the functional segregation of the entorhinal cortex. Although an oversimplification, one could perhaps say the medial entorhinal cortex is spatially tuned, whereas the lateral entorhinal cortex is non-spatially tuned. This suggests that rostral levels of area 29C may be non-spatially tuned, whereas caudal levels of area 29C are spatially tuned. In detail, the non-spatial information from the lateral entorhinal cortex can transfer to the proximal subiculum and then can travel further to the rostral part of area 29C. In similar fashion for the medial entorhinal cortex, the spatial information from the medial entorhinal cortex can transfer to the distal subiculum and then can travel further to the caudal part of area 29C. This projection about functional segregation of area 29C now only needs testing. Well, what we've been arguing for is that the amount of network knowledge is important for the level of detail at which we can explain network function. And although the current level of anatomical knowledge of brain connections is sometimes limited, a connectome already provides a useful knowledge structure that allows integration of information about brain structure and function. Here are some of our ambitions for the very near future. We are optimizing our database structure with the help of a bioinformatician. And when the new database structure is completed, we will add the hippocampal and parahippocampal connections for the monkey and human. As always, this is not a one-man or woman job, and therefore we would like to thank Menno Witter for all his guidance and anatomical knowledge, Jürgen Sugar for his great input on the retrosplenial expansion, Ingrid Riphagen for the wonderful help with the literature queries, and Arnold Kuzniar, who is currently helping us with database restructuring. And thank you for your attention.
Thank you so much, Niels and Natalie. I think that was very good. I have a question to us off with, but please go ahead and, oh, Giorgio is clapping. Thank you. I think we would welcome the questions from the group, so you can either turn on your microphones. It should be green if it's on, or you can type it into the chat as well. So the question that I have, it's kind of a larger theoretical question, is, and you started to allude to this, because if you are moving to the monkey and the human, then we have the possibility of getting some of this functional connectivity information from the MRI studies. Do you think that this inform the connectivity that you have from track tracing, and if so, how?
Well, I'm definitely attempting to go into the MRI with the human hippocampus. That's one of my current plans. And scan white matter connections at the highest possible resolution in post-mortem tissue, and then see if we can somehow compare connectivity seen in the human from MRI with that of what we have in our database. But that still has some gaps that we need to fill, but we're working on it.
Okay, great. So, Kristen, I am not going to try to pronounce that last name. I apologize. Has a quick question. How do we get the original articles from the database? Do you have a good way of doing that, or should I show them the NIF way?
No, we have no original reference in the database. You can search in the database by clicking on our website in reference search. Oh, nothing is happening there because we're still in the PowerPoint. Well, if you click on that button, you go to another website, and there you can look for connections, and there is a connection to PubMed, and there you can click on the link, and there you can find a PDF of your interest.
But since we do not have the license for all these articles, it's really dependent on your database access to the or not. We cannot redistribute.
Your microphone is now fading out a little bit. Yeah, I think this reference search, so that's good. You are aware that we also have your data inside of the NIF, because we've essentially taken it in order to compare to BAMS and some of the other sources. And we actually also have that information, the links to PubMed. So if, let me just set up the copy link address. Here we go. And paste. Nope, that's a JavaScript void error. All right, let me try that again. Okay, so there are a couple of different ways to be able to get that. Hopefully we can answer these questions. So Georgia was saying that the audio is way problematic at this point. So perhaps I should. Yeah, we can hear you, Giorgio. Yes.
Yeah, I missed the last answer out of that it was either garbled or bad stated. So I don't know if others are having the same problem or not.
Maybe could we discuss? Andrea said me too. Yeah, I think that the last time I think both of you were talking. Maybe I'm not sure, or maybe I'll just shut my my microphone off so you can answer that again.
Okay, we can answer it again if you want. If you go to our website, you find a tab with reference search. Niels is now trying to share our website. Yeah, and there it will come. One moment, and then you will see it. 1, 2, 3. 3, 2, 1. Yeah, there it is. There you have a reference search website on our website. You can look for the connection you want. You can choose either afferent or efferent, and then you can go for an injection area, for instance, CA1, and take a projection area, circulum, and you click on go, and then the projections will, you have to scroll down a bit. Then you will see all the papers which are in our website, and on the right side you find a link to PubMed, so you can go directly to PubMed.
And currently there are some duplicates in the search results, but we will be working on deduplicating this result, so you will only see each paper mentioned once for each connection.
Can you hear us now?
Oh, I think. Yeah, you're very clear now. I'm not sure what happened a little bit ago, but that's very good. Mikhail, do you have any technical questions? So Mihai is at the moment. No, we can't hear you. I'm sorry. You think you might have to turn on your microphone? So Mihai asked if we can hear him, and apparently we can.
And I have a question. I typed it up, or I can verbally, either way. Okay. I understand from the slides that you are considering as evidence track tracing like studies as well as intracellular injections. And I was wondering what the relative proportions are in the database, both in terms of absolute numbers and how many connections have some information on intracellular injections as well.
Now, can you hear me?
Yep, we can hear you now.
Okay. First, I didn't follow carefully the first part of the presentation.
Mihai says, okay, he will try to type it, I think. Because again, I think the sound is really bad for. Okay, so I think while he's typing, do you plan on trying to add a lot more data from the single cell tracing set? Oh. So Mihai is asking what are the plans of integration with BAMS? So are you planning to export this data to BAMS or export it to RDF? I presume that would be reasonable. Well, I think that's quite fair. I mean, certainly we are definitely in a brave new world in terms of making our raw data and the direct result of a lot of experimentation available to our colleagues. This has never really happened before. I mean, we used to have papers. We still sort of have papers, but now we have this whole new medium, and I think it would be it will require some thinking on all of our parts. Mihai is saying that would you like to add a plugin, temporal lobe, that does not export the data? Right, so Georgia, Menno is typing, but Giorgio, can you give a little bit of background of this meeting in Germany, because it might be a benefit to the group. I mean, we've obviously had various people to bring a lot of the experts together in various brain regions or various cell types, so that we can actually have a set of vocabulary for discussing some of these things, or at least this kind of Rosetta Stone type of approach. I'm very interested actually in looking at this a little more carefully myself. The Rosetta kind of structures that you have come up with. Obviously, you're not alone in bringing those together, and obviously others will, based on the species that they mostly study, would actually be color, sort of, would be a coloring of which structures they would potentially focus on and what kind of things that they would actually discuss in these Rosetta Stone-like structures. But this is a very interesting open area of research. So Mena said that please.
I think that the Germany meeting that they're referring to is the neuroinformatics meeting. There is something that is going on, which is people in my lab and this effort on the hippocampal, more track tracing level, as well as the effort on the brain region parcellation, also focused on the hippocampus that Mano collaborated with Yali with. There is some quite obvious synergy and complementarity there, and there are some initial steps taken in integration, such as using the same color scheme, hopefully using the same Rosetta Stone. But I think that there is a lot that can be done in terms of linking the databases and making them interoperable. And that is certainly not a matter of lack of will, but it does take quite a bit of work. So it's a matter of both resources and priorities. Certainly, I would agree with Niels and Natalie that there is an urgency in making the system robust and quality check. And I view the possibility of integration with complementary tools as a added on opportunity and truly be a great winner for everyone, for the community.
I fully agree on what you just said. So it's if I look at my own, or if we look at our own work, what we present now on the web and what we present now in terms of a PDF and review articles really is something that other people can explain, but a lot of this work is or can understand. A lot of this work requires such an extensive explanation. I think the way we described space
Oh, sorry, I just turning on my microphone because my Mihai was asking, what do you mean by raw data?
way, numerical way. If we would put that out on the internet, it wouldn't make sense because you would not understand the labels or what the numerical coding would stand for, and it would not be a simple one-on-one transformation that you could apply to that.
And on the other hand, all the connections which are reported on are included in our database, but we are not using them all to report on in the connectome PDF. So we already made a selection, and I think it's important that we annotate these
And
connections
curate, yeah.
and curate all these connections which are in the database very carefully. And I think at the moment we are not there yet. So I don't want to share this data to other people, because I think only people can make mistakes with it.
Yeah, it's important. It's important to understand that the connectome, the PDF diagram, is basically a cleaned out version of our database. That's one of our slides basically in the presentation that we just gave. How do we select what is a true connection and what is maybe non-information? How do we separate that? And in the database, everything is just there.
That makes a lot of sense. In the PDF, you are actually, in fact, releasing your data in its PDF form, and not the database form. But it is, in fact, you do share your data in a way on the internet, which can be looked at by lots and lots of people, and that's I think that's very beneficial to the community. I wonder for myself. What would be the sort of the timeline, I suppose, on getting more a richer set of data, including perhaps some of the strength information. I'm assuming that you have that coded somewhere in the database. You could correct me if I'm incorrect, but would you?
Definitely, we want to somehow visualize, but as said, it's everybody. Well, most. There's not an absolute system in which this was reported in the literature. So, what we could do is, discuss this with experts in the field, and we sure do, and then make a rating ourselves. But that means that we are distantiating us from the original publications, and we make an overview, and that's our overview in a way. So that's what we then have to offer as a sort of expert opinion on the connections. But yeah, we've had lots of discussions how to proceed with this project. I mean, we could attempt to try and put in all brain structures of the rat, and then come out with a review every year or so, and then be busy for the rest of our lives. Or we could try and see, well, what questions can we ask this database? What is it that we want to find out about? And well, one of my current interests really is to look up how we can use this information across species. How can we go from mouse to human, basically, and how can we do in a more tight way what people see in neuroimaging results with what we see in terms of connections in the database, and maybe these worlds will grow closer in some way by having that link. So that's
And then a prerequisite is that for the human data we get DTI data or something else, because the track tracing data in humans is of course very sparse.
It's very limited in
Right.
the amount of papers.
Yeah, that was my original question, because again, we have a lot of this DTI data that has been shared from the 1,000 Connectomes project and the new Connectome project from David Van Essen, and various other efforts. And actually, I just saw recently, I don't know if you saw this article, but someone was able to train a dog to sit in a scanner. I can't imagine you could train a rat to do that, but I think we're certainly getting a larger, species subset of this MRI ATI type data. So we would have the possibility to actually compare side by side, at least in some species, what would be some of the track tracing versus the functional connectivity information. So I think that really, we have a place where this is starting to possibly integrate. So, and I think you are actually thinking in a similar way, that this would be a very interesting thing.
Human track tracing in vivo, at least, is very different from what we have in our database currently, because the resolution of in vivo track tracing in humans is just poor compared to what we have in our database for other species. So the only thing we can currently attempt is to get better track tracing going in ex vivo human specimens, and that is what I'm currently attempting. And others are, of course, doing so too. So if we can make that step, then going from across species will be, I think that will be something that will become possible within the next next 5 years or so.
Yeah, that would be very cool. One other thing that you touched on, which perhaps you can address and maybe explore a little bit further, is Giorgio has raised his hand. Giorgio, did you want to ask a question before I ask my next question? Because I feel like I'm monopolizing.
No, I can go either before or after. I think there is time for at least 2 more questions.
Yep, we do.
Go ahead. Okay. My question is whether you expect a very substantial addition of data from efforts such as Partha Mitra's tracing, mapping that brain, which is the whole brain level, but certainly includes the hippocampus, or similar efforts that the Allen Brain Institute is putting forward using complementary techniques to that. Do you actually expect like a storm in a teacup, or like a doubling or tripling or a fold factor amount of knowledge for specifically the hippocampal formation?
It would be very nice if there was more information coming on of the connections of the whole brain. Of course, I mean there is already a lot of information, but if there is more and more, especially more detailed information about, especially the 3D information, that would be wonderful. I mean, the more you know, the more you can conclude about it. So
then again, you can do large-scale experiments, but if no one is looking into these results in a systematic way, then, or if there's a bottleneck in that sense, I mean, you can do.
I'm specifically asking about the. I'm sorry. I'm specifically asking about the raw data that will come out of this effort, and specifically for the hippocampal formation, so the area that temporallobe.com.
I'm afraid it was not really clear.
There's a really strong echo.
Yeah.
What Giorgio was asking is whether we expect something from the amount of raw data that are generated by those large connectome projects.
Well, if we have access to it and time to look into it, and I'm sure we can learn from that. But then,
Nano, can you turn off microphone? Speaking, I'm sorry, but I think that's causing the loop.
We completely lost the connection now.
No, I got the other microphone off finally, so now it should not cause the echo.
Okay.
we will draw data and multiply in the near future. I don't know.
No.
I don't know. Yeah. It depends on so many factors. If people are willing to share the data, if we can store it that's another major issue, of course. And then if we have true,
Yeah.
but if someone is producing a lot of experiments and producing a lot of raw data, doesn't necessarily mean that I have the resources to look at that. So there needs to be some sort of coordination on that level in order to be in order for that to work out. I mean, I would love to spend some of my time on going through raw data, but simply no one can afford, I'm sure, to make that a full-time occupation.
Yep, that's very true, and I think Menno is strongly thinking, so I think you were addressing the question, but I don't think it was read to all. So, strongly thinks that the connectome data need to be annotated by experts in order to be of use, and I also would tend to very strongly agree. Is saying that it's useless that unless someone is supported financially, right?
Sure.
Supported, financed by someone. Okay, I think I understand what that means, but I'm not 100% sure.
Well, I mean, it means that if there are experts in the field, like all researchers, we all need to work for our papers and do new experiments, write our new grants, and the higher you come up in the hierarchy, the more management you get. So that means that the less time you spend on looking at raw data, typically. If we're producing a lot of raw data and we're expecting that the community will just by itself start looking at it, then I'm not so sure if that will automatically happen. So some effort should be put into structuring the pipeline, basically, of making data and annotating it.
Yeah, and interpreting it, because that's also an important point.
Yeah, but I think I would potentially argue that you know, a lot of this raw data is being looked at by graduate students and potentially non-complete experts, but they can actually apply some interesting tools that maybe we would have not ever thought about to actually pull some things out of this data that potentially not every one of us or any one of the experts would be able to see. So, you know, raw data often has uses that are very unintended by the community, but it's nonetheless a very worthwhile thing to expose. And I think that a lot of these connectome projects do that and have plans. So Menno, again, he is writing that I do a lot of track tracing, and all those experiments are annotated and interpreted through the fact that I happen to have grants to do so. I cannot simply go into someone's raw data and annotate unless I have an experimental questions that I can address by using them. And that is actually a very, very good point. But you know, this is definitely going to be this. But, you know, people do have grants also to reanalyze existing data sets. Giorgio?
Yeah, my question was much simpler than that. Temporallobe.com did an outstanding and commendable job at taking a lot of papers out there, annotating them, making the resulting knowledge available in digital form. So this is truly a knowledge base. What I'm asking is whether the current efforts, such as the one going on with the transformative NIH support and the Allen Institute, if you expect that there will be a substantially greater volume of papers, which is your starting point for putting data onto temporallobe.com, or whether you imagine that would, you know, that what there is to be said about the hippocampus has been said, or that that those are too brain-wide in order to say something specific and massive about the hippocampus.
I think if you talk specifically about hippocampus, I think a lot of it is known about the projections. But on the other hand, we also do still have some questions about what are the neurotransmitters the connections are using. Is it glutamatergic? Are they GABAergic? For some of the connections we know, but for other ones we do not know. For instance, the CA2 area is sort of a mystical part of the hippocampus,
which is receiving more attention.
which is receiving more attention at the moment. But
but